Welcome, everyone. Welcome, welcome. We're going to get started here in just 1 second. As folks are joining us, getting logged in. Awesome. Welcome. All right, so it is the top of the hour, 2 o'clock where I am, 1 o'clock Central Time for our monthly webinar series. Thank you all for joining us today and welcome to the Monarch Conservation webinar series. Today's webinar is the science of radio tagging monarchs. Our presenters today are Ashley Fischer from the Xerces Society, Lee Brown and Helen St. John from James Madison University, and Sean Burcher from the Cape May Point Science Center. My name is Alyssa Taylor. I am a program coordinator with the Monarch Joint Venture. I am also joined by my coworker, Jenna Tots, who is the education coordinator. She'll be working behind the scenes on tech stuff. We want to thank our sponsor for their support for our webinar series. Our supporting sponsor is Reinvent Your Waste. We truly appreciate your support and dedication to monarch conservation. If you or your company is interested in sponsoring this or this year or next year, please feel free to email us at learn@monarchjointventure.com or .org, excuse me. Jenna will put that in the chat. Before we get started, we do have a couple of just quick reminders for webinars. So a couple of guidelines. If you're joining us today, but you didn't get a chance to pre-register, we'd love to count you for the attendance and also so that you can receive any of the follow-up information that we might have for our webinar today. So please feel free to email learn@monarchjointventure.org with your name, city, and state so that we can get that information to you. As a reminder, this webinar is going to be recorded, so if you have to pop off early, don't worry, it'll be recorded on and put onto our website by Friday of this week. So keep your eyes peeled for that. And then the entire Monarch Conservation webinar archive is on there as well, and free and available to watch. So there is about 10 years worth of Monarch Conservation webinars on there. So if you're looking for a particular topic, feel free to check that out as well. During this webinar, we're going to be using the Q&A box that should be found at the bottom of your screen. And we'll be using that to kind of collect the questions for our speakers at the end of the session today. So the Q&A box should be found at the bottom. If you can't see that, there's a little circle that has 3 dots. It says more. You might have to click that and then you'll be able to find the Q&A from there. Also, please feel free to use the reactions during the presentation. So remember, this is our webinar. Our speakers can't see you, but they can see the reactions that you might have. So I see them already popping up there. So those are also found at the bottom of your screen. And then we do want to remind you as well that we have translated captions and captions available on the Zoom call. So if you would like to add captions in a language other than English, click the arrow on captions and then set the translation button to the language of your choice. And then last but not least, our webinar speakers represent their own viewpoints, which are not necessarily those of the Monarch Joint Venture. So without further ado, our speakers today, Ashley is a conservation biologist for the Xerces Society, specializing in the overwintering monarch biology. She manages the overwintering habitat database and helps facilitate research projects working with the Xerces partnering organizations and universities. She also works with local stakeholders on the central coast of California to provide guidance in management and restoration of overwintering sites. Ashley holds a bachelor's and master's of science in biology from Cal Poly State University in San Luis Obispo. We also have Helen St. John as a current master's student at James Madison University studying monarch movement ecology under Dr. Lee Brown. She has a passion for conservation biology and hopes to continue future work in animal movement and applied ecology. She received her bachelor's in biology from Grinnell College. Lee Brown is an assistant professor of biology at James Madison University. Her conservation-focused research has centered around movement and demography of bird and butterfly populations in human-dominated and natural landscapes. She received her PhD at Stony Brook University and held positions at Harvard Forest, the University of Georgia, the University of Connecticut, and Tufts University before landing in Harrisonburg, Virginia, where she resides with her daughter, two conservation detection dog wannabes, which I believe are joining her today, and a mischievous kitty as well. And then lastly, Sean has a passion for solving difficult problems and enjoys learning new things along the way. He receives his PhD in nuclear astrophysics from the University of Tennessee while studying the origin of the elements in our universe. From there, he worked as a researcher in the Nuclear and Chemical Science Division at Lawrence Livermore National Laboratory, he is a senior R&D scientist at Cellular Tracking Technologies, where he designs, builds, and deploys cutting-edge wildlife telemetry devices. These novel tracking devices enable wildlife conservation research on every continent worldwide. Sean is also the science director at the Cape May Point Science Center, where he participates in many local conservation projects that help us understand and protect the wildlife of the Cape May area and beyond. So we have a lineup of fantastic speakers today. I'm going to cut off here and turn it over to Ashley to get started. Thank you all again for joining us.
Okay, sorry, it was a little tiny.
There you go.
Awesome. Can you guys see my screen?
Yes, it looks great.
Great. Okay. I'm going to get started. Thank you for the great intro. That are lightly separated by the Rocky Mountains and that little dotted line. Both of them spend their time in the spring and summer in the green and yellow areas on this map. And they spend that time focusing all their energy on finding mates, breeding, and laying eggs on milkweed. And because of this, all that energy is going into that. Adults actually only live 2 to 6 weeks in these summer months, which is a little bit different than their overwintering or migratory counterparts. Migratory monarchs, in contrast, are generally the last generation of summer or early fall, and the environmental changes trigger some physiological changes in them as larvae that help them emerge as adults as migrants that are then able to travel hundreds to thousands of miles to their respective overwintering locations. The eastern ones go to Mexico, and they're a larger population, where the slightly smaller population in the west travel to the overwintering sites along the coast of California. Both of these, all these migrants are able to live roughly 6 to 9 months, waiting out adverse conditions until milkweed reemerges and they return to breeding and lay that really critical generation in spring that starts the whole series over again. So I'm going to focus primarily on the West because that's what we're focused on. But I just wanted to compare Western and Eastern overwintering sites really quickly. While Eastern monarchs, it's a larger population. These overwintering sites start around like way higher elevations, like 9,000 feet. They are away from the coast and the OML fir forests of Mexico. And there are way larger population-wise and size-wise overwintering sites, and relatively fewer of them. So the populations are a lot more condensed. And in contrast, in the West, these overwintering sites tend to be way lower in elevation, generally speaking, pretty darn close to the coast. But it's a smaller population and it's way more diffusely spread out over hundreds of different overwintering sites. These span all the way from north of the Bay Area all the way down to Baja, California. And because there are so many, a lot of them are pretty close to each other. So because this population is way more spread out and it's slightly smaller, this is actually a pretty healthy sized population at an overwintering site in Morro Bay, California. Those dark spots are big clusters of monarchs. And generally speaking, they tend to arrive in October, with peak overwintering happening around Thanksgiving time. And by the time March rolls around, most of them have left this area, returned to breeding, and are starting to lay those critical spring generations that then migrate inland and back upwards to higher latitudes. But we really want to track these monarchs, which is why we're here today to talk about that. And it's mainly because there's still just so much we don't know. And this is about what maybe migratory corridors are most important, or that most of them use, or the exact timing and pace in which they travel. And if we could track individuals, we would be able to better identify these areas and then better protect them. And secondarily, like I mentioned, overwintering sites, which can be shown here in Morro Bay, my example area, in orange. A lot of times because there's so many sites, can be really close together. And we've noticed, thanks to our Western Monarch Count volunteers that go and monitor the numbers of monarchs at these sites throughout the season, that there are fluctuating numbers between these sites. Some of them are used regularly in really high numbers. Others are only used at the beginning of the season to be abandoned. And we know from other previous studies that use like sticker tags and stuff that there is some level of movement, but it's not really clear why they choose some sites over others, or how often they're moving versus how often they may be just dying or leaving the area earlier than we were typically thinking that they do. So being able to track them would help us figure that out. But as you could imagine, tracking individual monarchs is really hard because they're so tiny and fast. If you've ever tried to chase one as a child or as an adult, it is difficult. And in the past, the predominant method that has, been the basis of some foundational studies, we're using sticker tagging, which you can see here in this photo, what it looks like, where there's an individual ID that is then attached to that monarch. You can report where you recite them. But the issue with this is that you have to handle and tag lots and lots of monarchs just to get enough that you can happen to have the chance to recite them at an overwintering site. And the chances of you being able to see these tags as often as they fly by is pretty low because it's really hard to see them. For example, this is what a cluster can look like when it's kind of far away from you through binoculars, which is how a lot of these sites are being surveyed. So it's a really useful tool, but we're not getting the resolution or frequency of movements that we would really love to see. And that's why we're slowly turning to radio tagging and telemetry, which we think is, it's beginning to be used more often with monarchs, and we think it's the future of how we're going to better understand these movements. So how does it work and what is it? Radio tagging or radio telemetry is referring to basically the method for marking and tracking individual animals, where the tag itself is transmitting information basically through the air to the researchers. And what's really nice about that is that you really don't have to capture the animal every single time you want to figure out what ID it is, and you can get it from afar without having to handle them. And sometimes even be in eyesight of them. So this, because it's so effective, it's been used to study movement, migration, home ranges or territories, and habitat use of a bunch of different species of animals, like this ash-throated flycatcher here, and lizards and deer and wolves and a lot of different animals you could potentially think of. They've used this on because it's a really tried and true method that's actually been around for decades in various forms, though the technology has advanced a little bit. But it does work the same way, always, generally speaking. And it can really be broken down into a tag or a transmitter and a receiver and an antenna. So starting with the tag or transmitter, this tag is what you would attach to your study animal of interest after you capture them. And it itself has a power system on board that can then be used with the hardware to send out a burst of electromagnetic energy or radio waves that encode the unique ID at a certain frequency that the tag is designed to emit that signal at. So like different models of tags use, generally speaking, in the past, it was like one of two signals that they tend to use or capitalize on. And then also, depending on how it's programmed, in a lot of ways, the signals can be released at different time intervals. So you can release a little radio wave pulse, say every 5 seconds. So when you release the animal, you'll have those pulses being consistently released. And then the signals or pings can be picked up by a receiver with an antenna attached to it. And the antenna is shaped or designed in a way that is specific to that frequency of the tag, and then transfers that information to the receiver, which is kind of like the computer or processor. And then it will tell you two really important pieces of information. The tag ID, which tells you like, that specific tag ID that's associated with, say, Sheila the bird right here. And also the signal strength, which can kind of give you an idea of like whether, how close potentially that animal is to where you are detecting them. That being said, signal strength can be affected by a bunch of different factors, like how far away the animal is, the habitat, if there's a lot of trees in the way or something, tag type, antenna type and orientation, and of course, power levels of the tag. If a tag is battery powered and dying, it's going to be a weaker signal. So those are just something that help you interpret those signal strengths that you get. And moving away from my little PowerPoint cartoon, there are actually a couple of different receiver types that are typically used. Handheld radio telemetry receivers look like this. You can walk around with them, which is super cool. Drive around with them your study area. So it's really mobile, but unfortunately, us humans that have to operate them have to leave the field eventually, and we don't want to miss some tag detections while we're gone. So another different kind of receiver type that is popularly used is a remote station or remote tower. This is a temporary one, for an example, and these can be listening 24/7 with a big battery, and you can even hook them up to the cellular network so you can get your information on board while you're sitting at your computer. You can check and see what animals you've detected. These are all really cool, but traditionally in this sense, these are local to your study area if you're just using all the sensors that you're setting up and choosing to monitor in a certain area. So unfortunately, most individual research groups can't put out the hundreds of different remote stations that you'd need to study a really long distance migration. So people, especially bird people, migratory people, really thought about this, and specifically Birds Canada in 2014 came up with an answer to that, and that is the Modus Wildlife Tracking System. It's essentially a system or a network of radio towers that can be individually put up by anyone who wants to join all around the world. And the cool thing about it is that they're all basically reporting all their detections to the same database, which means that if you're a researcher that tags an animal in Canada and it flies in the range of your tower in California, they will get that information. And vice versa. So it's basically everyone pooling their resources to better understand these long distance migrations of animals. And they design them to pick up multiple different kinds of tags, as you can see with these different shaped antennas. Those are listening for different tag frequencies. And this is a good example of what a motor station looks like that was put up by my colleague Levi Souza of CDFW. So the U.S. Forest Service International Program gave Xerces and Point Blue Conservation Science grant money to essentially explore how the use of this technology that has been so successful with birds to how we can potentially use it on monarch butterflies. So naturally, our first goal is to figure out, okay, what kind of tag can we put on a monarch butterfly? And a little over a decade ago, tags were simply just too heavy to feasibly use them on monarchs. For reference, a penny is 2.5 grams and a monarch weighs about 0.5 gram. So that's a big ask to fit all that technology into such a small package. And like I said, around 2015, tags weighing 0.22 grams, so like almost half their body weight, were starting to be used. And then more recently, 0.13 grams with some success, but there's still a lot of concern that they were too heavy, or the battery had to be shrunk down to the point where it would really limit the tag's lifespan and ultimately the usefulness of it. Lee and Helen will speak more on tag choices and attachment choices later, but there has been work that has gone on in the background with some level of success using these kinds of tags or slightly more specialized tags. So in the beginning, we were leaning towards these low-tech nano pin tags. This is an example of one that we tagged a while back in a pilot study for CDFW. And it weighed 0.13 grams and the battery lasted a little over 30 days if you really stretch it out. But ultimately, the CTT cellular from Blue Morpho tag from Cellular Tracking Technologies, Sean and Lee and Helen will tell you more about. ultimately was our choice. And that was because it weighs 0.06 grams. So it was basically half the weight of that last other commercially available tag. And one of the ways that they've achieved this, thank you, Sean and team, is that it has no battery and it's just operating off of a little teeny solar panel. So that means it's sunlight dependent, but it would theoretically not die out, 30 days in. It could theoretically last the life of the Monarch or however long we could get it to stay on the Monarch. And a different other unique thing about them, though, is that it functions on the 2.4 gigahertz frequency to send that signal out, which can be really, really cool for a lot of ways that Sean is going to tell you about. That is totally beneficial. But one of the main drawbacks is that it was, at the time it came out, it was not compatible with hardware historically used in MODIS stations. So all those orange dots you saw of towers on the map would not be able to detect these at first. So I know that there's a lot of people working on basically getting these up to date, not just us, but part of our other project, or part of this project, now that we identified a beneficial or the best tag for them, is to provide the hardware to these independent researchers or organizations to retrofit their MODIS stations. So right now we're especially focused in California, where we know Monarchs will be. And our goal is to retrofit at least 20, and we're about halfway there right now. So last but not least, our last goal is to study overwintering monarch movement here on Vandenberg Space Force Base, which is nearby me. There's over 30 overwintering sites. I mean, either historically or currently used, which is marked in red. The outline is Vandenberg Space Force Base in blue. And we're going to be using these tags to track monarchs at 2 to 3 different sites, tag them, and then track them all over the base using all those different technological or receiver systems to see how they're moving in the winter and better understand what they love in a good overwintering site to better conserve this habitat. And that's about it. I just want to thank all our partners. I'm presenting a lot of work that a lot of other people had to do with. So with that, I will stop sharing.
All right. Thank you so much, Ashley. So this next part of the presentation is going to be done by myself, Helen St. John, and my professor, Dr. Lee Brown from James Madison University. And we're going to be taking you through the research that we've been doing these last couple of years to really confirm that the tags are safe to use on monarch butterflies. So we sort of had two big questions that we wanted to try to answer these last couple of years. Lee, could you go for it? Yeah, the first one is, which tag should we use? And in particular, are there some tags that are still too heavy for use on monarch butterflies, even if they're still really light? We need to make sure that they're not going to affect the butterfly's flight or how they behave when they're flying around. And then our second question is looking at which adhesive we should use to attach the tags. And the goal here is to try to use a glue that will minimize the impact on monarch survival while still maximizing tag retention. Because we don't want to use an adhesive that will just lose the tag, because then we put the butterfly through a process of tagging without getting any data and it was just useless overall. But we also don't want to harm the butterfly anymore by putting some bad chemical on them. And before we really get into these two studies, I just wanted to quickly thank all of our funders and our field team and research team from James Madison University, and of course, everybody that supported us by giving us access to their land or access to the tags, because we wouldn't have been able to do this research without them. Okay, so Ashley already went a little bit over the differences between the two tags that we know have been used on monarch butterflies. This first tag, the low-tech nanopin tag, that's been used in published studies on monarch movement for the last couple years. But then more recently, no studies have been published using the CTT tag, but we know that people have started to use them as they've become, as they've been released to the public. And just a quick note here that the low-tech tag pictured on the butterfly on the left is actually a 3D printed version that we used in our study. So that's not what the actual low-tech tag looks like, but it is approximately the same weight and size. And here are the main differences between the tags that Ashley already went over, but we're really interested in the weight differences between the tags, because at 0.13 grams, the low-tech nanopin tag would be to a butterfly as if we as a person were holding a 52-pound backpack proportionally to the average weight of human, while as the CTT blue morpho tag at 0.056 grams is more like a 22-pound backpack for a person. And so these weight differences, while actually are very small, are proportionally very large. And we needed to ensure that the butterflies were not changing their behavior because of these weights. Okay.
Thanks, Helen. So to test for the effects of the tags on monarch movement, we worked with breeding monarchs from a couple of farms local to our university in Virginia. And then we also went up to Cape May to work with migratory monarchs. And at both sites, we captured monarchs, weighed them, marked them just with an ID on their wing, recorded wing wear, wing size, wing damage, and then we tagged them with either the smaller CTT tags, the low-tech tags, our 3D printed version, or no tag at all. We then released the individuals in the middle of lovely fields full of nectar and milkweed. And then every 10 seconds, we recorded their behavior, something like this. If you can, oops, there's no sound, but. So that little ding, ding, ding is every 10 seconds we record what they were doing, if they were flying, if they were nectaring, if they were just resting, if they were, and where they were, if they were on the trees or what plant they were. And so what we end up with this is a movement path from which we can estimate move lengths, the turning angles, and the time it takes to make individual moves, which also includes resting, because if they're resting, it's how long they stay at one place before they go to the next. And then also the probability of resting. So in a really limited time, I'll share you a couple of the biggest take homes from this work so far, with the caveat that we are still waiting through some of the data, and so we might have some more insights for you when we get a little bit closer to publication, but we are pretty close. So with regard to move length, so just to show you here, there's a lot on this slide. So in this, on the left, these two figures are breeding monarchs, and on the right, these two figures are for our migratory monarchs. The top is just looking at move lengths, and the bottom row is just looking at the probability of resting. So with regard to move lengths, in breeding monarchs, we really saw no difference between the move lengths in the untagged butterflies and the CTT tagged butterflies, but move lengths were clearly shorter in the low tagged monarchs. In migratory monarchs, the difference in move lengths was marginally significant or not significant, depending on if you throw a couple other parameters in the model, between the untagged and the CTT tagged monarchs. But the movement lengths were again clearly shorter in low tagged monarchs. Also, just to note that the move lengths overall were shorter in migratory monarchs. So in the low tagged monarchs, breeding and migratory, the move lengths were similar, but even the untagged ones, they were shorter in migratory monarchs. At the bottom left, you can see the resting probability for migratory monarchs was much higher in the low-tech tagged monarchs than in the CTT tagged or untagged monarchs. And this was largely because a lot of these individuals sometimes just weren't moving much at all, or they would stop and then they would rest for a very long time. Some didn't really move at all. In the migratory monarchs at the bottom right, the difference in the probability of resting between the CTT tagged monarchs and the untagged monarchs was again marginally significant. And it was more similar between the low tech tagged and the untagged monarchs for the, between the CTT tagged and low tech tagged monarchs than it was for the breeding, in the case of the breeding monarchs. So our sort of biggest take homes right now are that we have pretty strongly concluded we believe that the low tech tags are indeed too heavy for monarchs. Some of the just overall. So at least our results now suggest that these tired monarchs are potentially spending a little more time initially adapting to tags. And the effect of the tags was a lot more variable, whereas in the migratory monarchs or some of the low-tech tagged monarchs would just take off, probably because these are larger, stronger monarchs, but others exhibited behavior similar to what we saw with the breeding monarchs.
So we think maybe these migratory monarchs need a little bit more time to recover from tagging, but it's worth noting, spoiler alert, Sean will tell you more about this, but there was, for instance, a CTT tagged monarch in Cape May that was detected over 700 miles away in Florida. So we really can detect long distance movements with these smaller tags. So in addition to the effects of tags on monarch movement, again, we need to make sure that if we're using these tools, the glues we're using to attach them aren't affecting survival. And these tags aren't cheap, so we actually want to make sure the tags will remain attached. So many researchers that we knew of at the time we started the study were using or intended to use super glue to attach tags. The published studies we came across were all using super glue. Eyelash glue and turbo bonding cement were being used by some other researchers and colleagues. And Vetbond, we decided to test because it's a medical grade super glue that has been used in bats. So we thought maybe if we find that super glue isn't quite good, like the right choice, maybe Vetbond would be better. So we reared over 470 monarchs in the greenhouse to test for the effects of these 4 different glue types on monarch survival and tag retention. And note, we only use the CTT tags for this because at this point, our flight path data was already suggesting that low-tech tags might be too heavy. We only had 23 monarchs in our first generation, so we had to mate them and got a whole bunch of monarchs. And it's really worth noting that the monarchs were able to successfully mate and lay eggs even when they had tags on. And we applied a small swatch of glue to the dorsal side of the thorax of the monarchs, of all of them. And for a subset of those, we tagged with the CTT tags to test for tag retention. And we would every day refresh the sugar water sponges in their cages. And we give them a light misting spray daily to mimic moisture that butterflies might be exposed to in the field. So the big take home here is don't use super glue or any glues with cyanoacrylates, which is the active chemical in super glues. Vetbond didn't even work to attach the tags, but we kept it in our study in case similar medical grade super glues are developed in the future that do work to adhere the tags. I say that, interestingly, one of our longest lived monarchs actually had a super glue tag, so it didn't go all of them, but the majority died within 24 to 48 hours. So you can see here, they're just much lower lifespan. Our control eyelash glue and the turbo bonding cement didn't significantly differ. And the eyelash glue and the turbo bonding cement, and the super glue all had a similar, the number of days until the tags was lost was similar. There's really big confidence intervals here because I think we only lost 1 tag or so with eyelash glue. It's 1 to 3. It was many fewer than with super glue and turbo. And we lost too few tags really to see a significant difference in the probability of losing a tag between eyelash glue and super glue or turbo bonding cement, but we lost fewer with the eyelash glue. So again, don't use super glue. And while not many tags were lost, we think eyelash glue probably is the best for tag retention. And it was also easier to apply.
All right, so just a couple slides to conclude this part of the presentation. In terms of our specific studies, of the 2 tags we tested, very obviously the CTT Blue Morpho tag is the better tag for use on monarch butterflies. And the eyelash glue is probably the best glue that we've tested to attach the tags. But we do want to just mention some broader tagging project conclusions. We firmly believe that when you are going to tag a butterfly, your research question and study goals should be clearly defined before you go to tag the butterfly, because you want to make sure that tagging is actually necessary for you to answer your questions. Because otherwise you might be tagging unnecessarily, and we don't want to do that. We also just want everybody to keep in mind that we should always be striving to use the smallest available tag that can help us achieve our study goals. Especially as in the future, more and more smaller tags are developed. And then, of course, before tagging, you should always carefully consider any unanticipated impacts of tagging, such as the handling time or the glue that you're using, or which stage in the monarch life cycle you are planning to tag. Next slide, please. And just as a quick example of another study that we're currently doing that requires tags, we are trying to relocate individual butterflies throughout the day during the breeding season, because we noticed last year that they were using trees a lot more than we expected. And that's very interesting because breeding habitat conservation for monarch butterflies really only focuses on milkweed and nectaring plants, even though we know that trees are important during the migratory season and overwintering. And so we wanted to sort of quantify the frequency that butterflies are either in the tree line or in the shade of the trees during the day or at night when they're roosting, or out in the open field throughout the day, which will hopefully give us a better idea of how important trees might be for the breeding monarchs. Next slide, please, or click a little bit. Yeah, so in the trees or in the open field. And then the last little bit that I just wanted to go over quickly, because I'm sure all of you were wondering about this, is what actually is the process of putting a tag on a butterfly. And so just very briefly, we start with a butterfly in your hand. Next click, please. And then in order to protect the wings, and especially the scales on the wings as we're handling the butterfly, we found a lot of success putting glassine envelopes over the wings so that as we're manipulating the butterfly, the scales are not being lost. Next click, please. And then what we have found to be really successful is to use sort of a makeshift pinning board that can hold the butterfly's body as we sort of place the wings down on either side with weights like washers or microscope slides. That way the wings are not in any danger of getting glue on them as you're applying the glue and the tag. And then the last step is to, like Lee said, it's really easy to put the eyelash glue on the butterfly because you can, click next, Lee, because you can sort of brush a little bit of the glue on the butterfly and on the tag itself and then wait for the glue to dry, which on average has taken us about 7 minutes. But that can vary a lot depending on the humidity or the temperature outside. But I can answer more questions about the tagging process at the end. I think it's now time to move on to Sean.
All right. Yeah, thanks, Helen and Lee and Ashley for the introduction. I'm going to talk about another aspect of this project, and that is Project Monarch. And Project Monarch is, well, it started as a project to study the movement of specifically the migratory generation of monarchs by the Cape May Point Science Center in partnership with Cellular Tracking Technologies. And it's now kind of evolved into a little bit of a collaboration. More than a little bit. You just heard from 2 of our collaboration members, the Xerces Society and the team at James Madison University. And we have all these other members now, and they're all deploying tags through our Project Monarch app throughout North America, which has been really cool to see. We're about 3 years into the project now. And at the heart of the Project Monarch, the way we gather our data is the CTT Blue Morpho tag and our Blue Series tags in general. And the fact that they transmit over this different frequencies, so the 2.4 gigahertz, which as Ashley mentioned is the same frequency as Bluetooth. And so that gives you more options for picking up the tags. So in the radio telemetry introduction, you heard about having these specific receivers with the antennas dedicated to the frequency. Since we're transmitting over the frequency of Bluetooth, pretty much everybody is walking around with a receiver in their pocket. And so we just needed to be able to turn them on, essentially make them listen for the tags. And so that's what the Project Monarch app does. And then I'm also going to talk a little bit about the Terra device. And Terra is primarily a device made for listening to bird calls. It uses AI to then identify the bird based off the acoustic monitoring. However, it also has for the 2.4 gigahertz tags, the Blue Series tags, like the Blue Morpho, and 434 megahertz tags. So it's a mini MODA station as well. So the Project Monarch app is free to download on iOS and Android. And the goal of it is to crowdsource the tracking of radio tag monarchs. And so as I mentioned before, the app allows your phone to detect these monarchs. And then it uses the GPS location from your device to acquire a location, and then it uploads to our server the time and location and the unique ID of the tag that you just detected. And as it's a really cool tool for gathering data and, you know, allowing members of the community to go out and do it, but it also is a really cool tool for promoting public engagement with monarch conservation efforts. We do it at the Science Center, and a lot of our other collaborators do this, where they hold public tagging demos. That's nothing new in the monarch research world. You know, people have been doing that with sticker tags for decades. But at our public tagging demos, where we put out some of the blue morpho tags, everybody shows up, you download the app, and then everybody gets some detections, and you just kind of, you know, it's a great way to get the, you know, conversation started about monarch conservation efforts in general. So a little bit more about the app. This is what it looks like when you download it. And when you're scanning for detections, you kind of see that second screen there. So in this case, we're detecting a butterfly called Kashi. So you get to name all the butterflies, which is pretty cool. We name them after, you know, people who have been influential in like the Monarch Monitoring Project or from the community in Cape May. Well, you can name them whatever you want. We've deployed a lot, so we've we've had a lot of interesting names. And then the next screen here you see is looking at the data from an individual monarch. This one's named Sam. And you can see this monarch was detected 5,563 times by 33 community scientists. And you can see on the map down there, its latest location, which is the little butterfly icon. And then the blue dots are the locations of all the other detections. And then all the way to the right here, this is one of the, you know, aspects that I really think drives engagement with the app is we have a leaderboard. So we've kind of gamified it. You score points for, you know, for detecting monarchs. So you can see our leaderboard here with a couple people. You can see Mark Garland at the top. He kind of heads up our tagging efforts at the Cape May Point Science Center. So it's no surprise that he gets the most detections. But we also have leaderboards for the most number of individual monarchs. And it's just a cool, fun way to, you know, to kind of incentivize people to get out there and scan for monarchs. So our stats on what we've gotten from the app so far. So user accounts created, we've had 6,256 users create accounts on our app. All of our collaborators included have deployed 354 tags and uploaded a total of 359,000 detections. So it's a lot of data. Here is the last location of all monarchs from the app detections only. I'm going to talk about detections from other sources in a minute. But as you can see, we're spread out throughout North America now, which has been pretty cool. The first year it was almost just Cape May Point, but this has really, really expanded. And we have people deploying tags all over the place, which is awesome. Here's some of the data, kind of the localized data, which is mostly what we've gotten from the app so far. You know, it requires somebody to be scanning actively, and so maybe the word's not out. You know, 6,000 people sounds like a lot. It's 6,000 extra receivers, but you know, a lot of them are concentrated in the places where these efforts are occurring. And so on the left here, we have all the monarch detections in Cape May Point, New Jersey in 2023. And you can see some hotspots here. The one towards the bottom right is the Cape May Point Science Center. So that's where we tag. So there's a lot of detections there. But then some of the other hotspots you see throughout town are locations where there are great gardens, you know, with a lot of flowering plants that the monarchs are nectaring on. Or a couple of them are roost locations where a lot of them, you know, are sleeping overnight before they have to cross the Delaware Bay. So for those of you who aren't familiar, this is the very tip of the New Jersey Peninsula. And so as they travel south, they kind of stop over here in Cape May Point before they have to make the cross over the Delaware Bay, which is about 17 miles. So monarchs tend to hang out in Cape May Point for a little while. In this year, we had at least one monarch that was detected in Cape May Point for 14 days straight, which was pretty cool. On the right here, you can kind of see the data you get from an individual monarch. This one was actually named Xerces in honor of the Xerces Society. And we see that, you know, through the detections where this monarch, you know, this was over a few days, but where the monarch moved through Cape May Point, which was pretty cool to see, to track its movement over a few days. It's kind of stuff that would be very difficult to do without, you know, with the old sticker tag method, but is possible with the radio telemetry method. And all this data that you're seeing here was crowdsourced from the public. So it didn't require, you know, scientists to go out and follow these monarchs around. You kind of just get this data for free after you put the tag out in the wild. So One of our other sources of, of Monarch detections now is the Terra device. And so this map you're seeing here is actually the live map from Terra's website. So if you go to that URL at the bottom there, map.terralistens, you can all see this map right now, and you'll see bird calls coming in. On this map, the yellow dots are the locations of MODIS stations, similar to the map that Ashley showed earlier. And then you see the Terra devices are the little, the green chunks there. And you see some Monarch icons in different places. And so the Terra knows about the Monarch tags. We have a list of the Project Monarchs deployed tags loaded into the Terra. And so it's looking for the latest location that a Terra detected one of these Monarchs. And you'll see one of those Monarchs is down there in Florida. And that's the Monarch we're going to talk about next. But before I move on, I want to point out at the top here, I have it circled in red, and the installed stations is a little over 1,000 right now, but our radio tag detections are already at 500,000. Now, there are a lot of Terras near this CTT offices, so that number gets inflated a little bit, but the power of this tool, as it gets more and more out in the wild, is just going to supplement the MODIS network and help detect not just the Blue Series, but also the 434. So at almost the very end of the migratory season for us in New Jersey, we tagged a butterfly called Lionel. It was early November. And it was picked up on a Terra device in Florida, which was about 726 miles away from the Cape May Point Science Center. And so that was our first documented, truly long distance migration with a CTT tag on. So this was like the confirmation for us that it is possible for, a butterfly to make it long distance carrying one of these tags. So Project Monarch in the future, one of the things that we're really working hard on this year is to kind of pipe all the data from these different sources back into the Project Monarch app to make it available for researchers and for members of the public. And so I've kind of put circles around all these different things, and the circles kind of represent like the detection range, and they all have their pros and cons. And so the Project Monarch app is a relatively short detection range. It's about the Bluetooth range of your phone. Think about staying paired to like a Bluetooth speaker or something like that. And it also requires active scanning, so it's not always listening. However, it's like a really powerful tool to drive engagement, like I talked about before. And the Terra, it's got a little bit longer of a range because it's got a bigger antenna, and it's passively listening. So as long as the Terra is on, it's always listening. It also has the live map aspect, so there's a little bit of engagement. You can log in, if you have a Terra at home, you can log in and see which radio tags your Terra has detected. But these are really concentrated in populated areas, right? You're going to find these in people's yards in the suburbs and stuff like that. And so that's where like the CTT sensor station and blue receiver and these that Ashley was talking about earlier, these are long range. You can set up these antennas where you're going to get a couple kilometer, range. It's passively listening again, so listening 24/7, and yeah, be installed in remote areas. And it'll also report the sensor data. So the blue morpho tags do also transmit temperature and the voltage on the solar panel. So that can be mapped to how much light the tag is currently in. And so next, I want to talk about something that we were able to pioneer last year. And so we kind of figured out how to do this at the very end of the season last year, and we only were able to tag 3 Monarchs with this new type of programming on our CTT Blue Morpho tags. And again, I want to highlight this is the first time we've been able to talk about this publicly. We wanted to make sure we had all our ducks in a row before we were able to talk about exactly what we were doing here. And we're leveraging existing crowdsourced location networks. They exist, a lot of large tech companies have them, but essentially what we can do is we can program our Blue Series tags to conform to the existing network protocols of these location networks, and then you can retrieve the data from that corresponding service. And so this takes it from having to actively scan with the Project Monarch app to there are already hundreds of millions of devices that are passively listening. However, again, they're concentrated in populated areas, so you still want the MODIS network and the towers to kind of supplement that. But this is what Lionel was one of the butterflies that was wearing one of these special new tags. It's the same tag, new programming. And this is what Lionel's track looked like, the detections from these existing crowdsourced location networks. So we got 191 detections in between the Cape May Point Science Center and the Terra in Florida. And this was over 28 days. And Again, 1,100 kilometers, 726 miles, but this is the most detailed, Monarch track over this distance ever recorded. It's not just point A and point B. We had 191 points in between. And so this really allows us to ask, to use these tags and these data sources to ask totally different questions that were just not possible before. And that's looking at the fine scale movement data, for monarch butterflies over these large scales, but identify stopover habitats and see where they're moving around, correlate movement data with weather events, and even, on the non-migratory generations, maybe study the dispersal of them. And I wanted to show this slide too, which is just the start of the data analysis on stuff like this. And this is what Lionel's track looks like if you color each segment of the track with the speed that it was traveling. And then there's a histogram here on the left where you see the speed in kilometers per hour. There were about 2 segments there in North Carolina where Lionel was traveling almost 80 kilometers per hour, which is pretty cool. It's very, very fast for a butterfly if you think about it. And so, this year we plan on, we'll be deploying lots and lots of tags with this kind of capability. And I'd showed that slide before, but I want to add a third one here. We're working on connecting all the pipelines and on our server side to be able to get all this data fed into the Project Monarch app and kind of have this as a central location for all the data, but also make it a public engagement tool. And we hope that other Monarch researchers, as has been happening in the collaboration, but continue to adopt the Project Monarch app and use it for their scanning and their surveying. If you wanted to go survey, like an overwintering site, like Ashley was talking about earlier, you could go to the site and stand in the center and scan and see if any of your monarchs are present, which is just, it allows the members of the public to do it and not, you don't have to have this dedicated infrastructure like the receivers and stuff like that. And so once we get all these data sources connected, we're going to get a truly unprecedented level of Monarch data available for researchers and for the public. And it's going to be really cool. Really excited for this year's deployment. And with that, I want to thank, the Project Monarch collaboration and Cape May Point Science Center. And yeah, we're looking forward to 2025. It's going to be our biggest year yet.
Thank you so much to all of our speakers. We have a lot of questions. So we're gonna be asking some of the questions today. We will likely not get through all of the questions that have been asked today, so just keep that in mind. But we will probably be sending off some questions to our speakers, and that will be included in our follow-up email out to everyone. So just as a reminder, again, if you want to receive that follow-up email and you did not register, to please email us at learn@monarchjointventure.org with your information. So I'm just gonna kind of go through and pick and choose some good questions based off of ones that have been asked kind of repeatedly. So there's a couple big questions that have come up a lot, and I think I will likely direct this towards Sean and maybe Ashley, since you've been doing some of the retrofitting. A lot of curiosity about MODIS towers. If an organization has a MODIS tower and they'd like to retrofit it to detect these tags, is that something that's possible to do? And is there value in that at this time to help pick up these potential tags that could be moving through if they're not based specifically in New Jersey, or if they're not based specifically in California, or if they are? What thoughts on that? Ashley?
I'll take it. And then please add on, Sean. So what's really cool about the MODIS stations is that you can go to their website and see who the owner of that station is. So, and it should theoretically have contact information. So if you're thinking of one in particular that you want to retrofit, that's how you get in contact with the person that owns it. And then you'd have to buy some of the retrofit materials to do that. But like, Sean, please hop in if you know of other like research projects, but I think it's like Lee and Helen, yours. I think there might be a certain Audubon society, but I don't think they're using CTT blue morphos. And then we're using them on the central coast of California. Maybe one other place in Idaho is considering using them, but I'm not really sure, and I think it depends on the location. So like for our season here in California, people surveying on the central coast, we will be telling them to at least download the app to scan for them. And we have been working on retrofitting those towers, but depending on where you are, it might be a little early, but no problem in getting started soon because it takes a while to get your stuff together and deploy it.
Yeah, absolutely. Just to add to that, we are well, there are collaborators deploying, Project Monarch tags or CTT blue morphos throughout North America now, and we have some inquiries from other places that weren't on that map yet. So this year it may kind of expand. So you never know if you get detections. And then we do have, at CTT, lots of people are buying our Blue Series tags, whether they're the blue morphos or the blue bats, which are just the battery powered version of those, and deploying them in all kinds of species. One of the other big projects that's going on now is studying hummingbirds. So there's a number of hummingbirds flying around North America right now carrying CTT blue morphos. So your MODIS station may pick up one of those. One of the cooler tracks from that was one was deployed in Alabama, and using the same kind of method I showed for Lionel there, where you get the pings off of all the phones. It was tracked all the way up to Alaska, to Juneau, Alaska. So that was a really cool, that was a Rufus hummingbird. But anyway, there's a lot of hummingbirds flying around. We'll have a lot of monarchs flying around this fall, and plenty of other species. So there's definitely benefit in updating your, retrofitting your MODIS station for Blue Series tags.
Awesome, thank you. So as far as kind of the phone app and MODIS towers and even the Terra, what is the distances that that tag would need to be from those in order to be detected? Right. So Ashley had it in her slide about the range of detection. There's a lot of factors in there, right? And so you have the complexity of the environment, kind of the how much vegetation is, even like the elevation of a tag. So a high flying butterfly, you're going to get a better range out of. But in general, a rule of thumb with phones, and again, it depends on the phone. Not every phone has the same Bluetooth antenna, but about 100 meters. And if there's anything in the way, it goes less. So 100 meters line of sight. Any sort of elevation changes or vegetation can decrease that. And then Terra is, we say, about closer to 500 meters, just because it has a bigger antenna in it. And if you mounted the Terra on the top of your house or something like that, it could be even more than that. And then MODIS stations typically, and this is actually using the same antennas that Ashley showed in her picture, where it was like a cross, and there was 4 little panel antennas, one at the end of each one of those points. We did a test with those on the roof of the Cape May Point Science Center, so it was about 50 feet up, and we were detecting drones up to 2 kilometers away. So we flew a drone around carrying the tags to measure the detection range. So that's kind of where they all fall in the detection range. Awesome, thank you. So this is more directed towards Lee and Helen. As far as was there a weight threshold that does not impair the Monarch's flight?
So I'll jump in, Helen. So we actually only tested the CTT tags and the larger low-tech tags. We really wanted to like 3D print tags of all like different ranges, but that just was outside of our time to really get enough a large enough sample size to really distinguish between the low-tech and the CTT tags. Largely, the ones with the CTT tags seem to be flying, like we didn't see any difference. And even now this year again in the with the breeding monarchs that Helen is following, we go out there, Helen can jump in, but they're flying around just as fast and crazy as the untagged ones. And as far as the migratory ones, I mean, those subtle differences, again, the move lengths were shorter overall, and so you're not going to see as much of a difference even between low tech and untagged ones. But the short answer is we didn't quite test that. The longer answer is definitely not low tech, and CTT is probably okay. I think the one thing that is we really emphasize, so we always made sure to tag individuals that had no or very low wing damage. And we often would go back and look at like the veins in the wings, make sure they weren't broken, like that top vein. If that's broken, that can really affect them. And this is why we also really encourage people, like there are unanticipated impacts, and any butterfly that goes through like, you know, a hurricane or something, it's going to get wing damage and it's going to have a hard time flying anyway. And in that case, any extra weight might impact it. That doesn't mean these tags, I mean, I think they're great for answering some of these really important research questions that we can't really answer in other ways very feasibly, with regard to conservation.
That's awesome. Thank you. I'm gonna ask like two more questions because we had a list. I know we're right at the hour. But somebody had asked a question as far as like the time it takes for you to apply the tag. And I think you were talking, Helen, I think you were talking specifically about the letting it dry and then you apply. And the question was, could you use an accelerant to like speed up the setting of the eyelash glue to like decrease the time?
So the eyelash glue does not have an accelerant that, or well, I mean, I suppose we could try to like find another chemical to mix it in, but then we'd want to do some more tests to make sure that that chemical accelerant is not affecting the butterfly in any way. There are some other adhesives that have been used on other animals that we did not include in our study. I know that there's like dental adhesives that are UV activated, so you have like a little UV gun that can like quick dry, quick harden the glue, and those have been used on things like oysters and mussels in aquatic environments. But as far as I know, no one's tried to use them on monarchs, and we didn't test them. But it's possible that that could work. We just need more tests done on that sort of thing.
Awesome, thank you. All right, and then last one, and then I'm just reminding everybody, we'll send these questions out to our presenters today to help get some of these other ones answered and some of the things that are a little bit more specific to different folks. But last question is just I think probably towards Sean. What is the lifetime of the tag? Like how long will that tag last, and could you potentially collect and reuse the tag after the monarch has died?
Yeah, so the tag is solar powered, right? So there's no battery. So as long as the tag's hardware itself doesn't get damaged, it will last indefinitely. And yeah, we have recovered tags off of dead monarchs that are still working. So yep, totally possible.
Awesome, thank you. Ashley, I see you're unmuted. Did you have one other thing to add?
I just wanted to say one other thing.
Yeah, definitely.
It's all super exciting, super cool. I just wanted to remind, just in case people are super excited about it and want to buy a bunch of tags, especially in California, you have to have a permit from CDFW to capture and handle monarchs in the state. So just a reminder that you'd have to apply for a scientific collection permit to do that. And now we're all good. That's all I want to say.
Yes, that's a great point. And then I'm going to add this question into, I know what time we're at. But there was a question of just like, how expensive are these tags? Like, what is the cost for the tags at this time, since this is fairly new technology?
Yeah, so the tags retail $175 a piece, but there's discounts in quantities. So, you know, when you get up to larger quantities, there's discounts. That's all available on our website, celltracktech.com.
Awesome, thank you. All right, well, we will collect a bunch of resources, the websites that were kind of shared out today. But I do want to just say thank you again to our presenters. Thank you for hanging on for a few extra minutes to answer some of our questions. Really cool things coming out as we get new technology available to answer some questions. So just a reminder for folks, Jenna just put into the chat there, we do have a survey for you to take, feedback for our webinar series, topics that you might be interested. Thank you all again for presenting. Thank you all for joining us and providing us with those insightful questions. And please make sure to fill out that form. Our next webinar will be on Tuesday, August 26th. And that will be building a framework for national native seed strategy in Canada and incorporating a two-eyed seeing approach into the Southern Ontario seed strategy. And we'll be joined by Carolyn Callahan from the Canadian Wildlife Federation. So thank you all again, and I hope you all have a great day, and thank you again to our speakers.